Assalamualaikum and good morning to each and everyone. My name is Shaira Abobakar and I will be the one who will be presenting from this video. So the content of this PowerPoint is to have an overview of transcription in both prokaryotic and eukaryotic cell, and to know the green cell, the long-range cRNA movement, and to know the crispy R in other non-coding RNAs. And here is the table of content. The first one is the transcription in bacteria. The second one is the transcription and RNA processing in eukaryotic cell. The third one is the crispy R, non-coding RNA in bacteria. And the fourth one is the other non-coding RNAs. So let us start with this slide. So this presentation explores the process of transcription in bacteria that is focusing on RNA polymerase structure, promoter recognition, transcription initiation, elongation, and termination mechanism. So in transcription in bacteria, it is a single type of RNA polymerase, and the core enzyme composed of 5 subunits, and it requires sigma factor for specific promoter recognition. So the bacterial RNA polymerase binds to DNA to synthesize RNA. However, sigma factors are essential for directing the enzyme to specific promoter site. So if the core enzyme is purified from bacterial cell and added to a solution of bacterial DNA molecule and to a ribonucleoside triphosphate, the enzyme binds to the DNA and synthesizes the RNA. And the RNA molecules, it is produced by a purified polymerase. However, these are not the same as those found within cells because the core enzyme has attached to a random site in DNA, site that it would normally have ignored in the IDO. If, however, a purified accessory polypeptide called sigma factor is added to the RNA polymerase before it touches to DNA, the transcription begins at selected location. And the attachment of a factor to the core enzyme that is increases the enzyme's affinity for promoter site in DNA and decreases its affinity for DNA in general. As a result, the complete enzyme is taught to slide freely along the DNA until it recognizes and binds to a suitable promoter region. Another one is the crab claw-shaped molecule and the positively short internal channel and the those script downstream DNA duplex. So meaning to say the enzyme structure that is resembling a crab claw is enable it to secure a DNA and separate strand for temporal accessibility. The enzyme then separates to the two DNA strands in the region surrounding the strand site. So the complex of the polymerase or the sigma factor and DNA with the strand separated is called the open complex. The strand separation makes the temporal strand accessible to the enzyme's active site, which reside at the back wall of the channel. So initiation of the transcription appears to be a difficult that is undertaken because an RNA polymerase is typically make several unsuccessful attempts to the assembly of an RNA transcript. And once about 10 nucleotides have been successfully incorporated into a growing transcript, and the enzyme undergoes a major change in conformation and is transformed into a transcriptional elongation complex that can move processively along the DNA. It is also located upstream of transcription start site at positive 1. The key sequences, which is the 35 element, which is the TTGACA, and the 10 element, the TATAAT, or the Pribnow box. So that means that the promoter region contains a conserved sequences that help RNA polymerase locate in a transcription start point accurately, and the nucleotide at which transcription is initiated is denoted as positive 1, and the preceding nucleotide is as a negative 1. So those portions of the DNA preceding the initiation site toward the tree and of the template strand are said to be the upstream from the site, and those portions of the DNA succeeding in it or the toward to the 5 end of the template strand are said to be the downstream from that site. The analysis of the DNA sequences just upstream from a large number of bacterial genes that it revealed that two short stretches of the DNA are similar from one gene to another, and that one of these stretches is centered at approximately 35 bases upstream from the initiation site and typically occurs as the sequence of TTG-ACA. And this TTG-ACA sequence is known as the 35 element, and it is also called a consensus sequence, which indicates that it is the most common version of a conserved sequence, but that some variation occurs from one gene to another. And the second conserved sequence is found approximately 10 bases upstream from the initiation site and occurs as the consensus of the sequence TATAAT. And this site is the promoter that is named the 10 element after its position or the Pavlov box after its discovery. And it is responsible for identifying the precise nucleotide at which transcription begins. And we have also the row-dependent or the row protein separate RNA from the DNA, and the row-independent, which is the hairpin loop formation that causes the RNA to release. So it is saying that the transcription concludes through either raw protein activity or intrinsic terminator sequence that is forming destabilizing structure. Just as transcription is initiated at specific point in the chromosome, it also terminates when a specific nucleotide sequence is reached. In roughly half of the cases, a ring-shaped protein called RO is required for termination of bacterial transcription, and the RO encircle the newly synthesized RNA and move along the strand in a 5'3" direction to the polymerase, where it is separate the RNA transcript from the DNA to which it is bound. So in other cases, the polymerase stops transcription when it reaches a terminator sequence, and a terminator sequence is typically folded into a hairpin loop that causes the RNA polymerase to release the completed RNA chain without requiring an additional factor. Okay, so let us now proceed to the transcription and RNA processing in eukaryotic cell. So we have three nuclear RNA polymerases, the polymerases 1, the polymerases 2, and the polymerases 3. And each synthesizes specific RNA type, for example the mRNA, rRNA, and tRNA. And the plant have a additional non-essential polymerase, and the prokaryote have a single RNA polymerase. So the eukaryotic transcription involves specialized RNA polymerases that is highlighting a sharp distinction from prokaryotic cells, which have only one polymerase. And the eukaryotic cells have three distinct transcribing enzymes in their cell nuclei, and each of these enzymes is responsible for synthesizing a different group of RNAs. Plants have two additional RNA polymerases that are not essential for life, and no prokaryote has been found with multiple RNA polymerase, whereas the simplest eukaryote or the yeast have the same three nuclear types that are present in mammalian cells. And this difference in the number of RNA polymerases is another sharp distinction between prokaryotic and eukaryotic cells. Another one is the structural similarities between archaeal and eukaryotic RNA polymerases. Multi-subunit structure conserved across domain, and the yeast RNA polymerase 2 has a 12 subunit, and evolutionary conservation revealed by high-resolution studies. So meaning to say, the archaeal and eukaryotic RNA polymerases each share the evolutionary root with homologous subunit and conserved core structure that is reflecting their close relationship. The surface structure of RNA polymerases from each other of the three domains of life, the archaea, the bacteria, and eukaryote. Several features are apparent from a close examination of this illustration. It is evident that the archaeal and eukaryotic enzymes are more similar in structure to one another than the other bacterial and archaeal enzymes. These features reflect the evolutionary relationship between archaea and eukaryote, and the subunits that make up each of the protein are indicated by a different color, and it is immediately evident that RNA polymerases are multi-subunit enzyme, and those subunits of the different enzyme that are homologous to one another, and it is shown in the same color. Thus, it is also evident that the RNA polymerases from the three domains share a number of subunits. Although the yeast enzyme has a 12 total subunits and 7 more than its bacterial counterpart, and the fundamental core structure of the two enzymes is virtually identical. In this evolutionary conservation RNA polymerases structure is revealed, it shows the homologous region of the bacterial and eukaryotic enzyme at much higher resolution. Next one, the eukaryotic transcription relies on transcription factor, and the primary transcript or the pre-RNA undergo an extensive processing, and the processing include the splicing, capping, and tailing, and the small RNAs and associated protein assist in RNA maturation. So, meaning to say, the eukaryotic transcription and RNA processing is involved in the complex machinery, including the transcription factors and specialized processing steps to produce a functional RNA molecule from primary transcript. So, our understanding of transcription in eukaryotes was greatly advanced with the 2000 publication of the X-ray crystallographic structure of yeast RNA polymerase II by Roger Kornberg and colleagues at Stanford University. As a result of these studies and those of other laboratories in subsequent years, we now know a great deal about the mechanism of action of RNA polymerases as they moving along the DNA, the transcribing of a complementary strand of RNA, and a major distinction between transcription in prokaryote and eukaryote is the requirement in eukaryote for a large variety of accessory protein or transcription factor, and this protein play a role in virtually of every aspect of transcription process, from the binding of the polymerase to the DNA template, to the initiation of transcription, to its elongation and termination. All three major types of eukaryotic RNAs, mRNAs, and rRNAs, and the tRNAs are derived from the precursor RNA molecules that are considered longer than the final RNA product. Let us now proceed to the another content of this presentation, the green cell or the long-range cRNA movement. So the cRNA movement depends on DCL4 enzyme, and it diffuses passively over 10 cells. The RDR6 amplifies cRNA by synthesizing the dsRNA. So, meaning to say, the cell-to-cell movement of the cRNA appears to depend on a specific dicer molecule called the DSL4, and the cRNA molecule produced by the DCL4 can spread over 10 cells away from the initial site of production. The movement is apparently mediated by passive diffusion, so the concentration of the cRNA molecule will gradually decrease with increasing distance from the source of the diffusing molecule that is become diluted. However, the plant also contains a RNA-directed RNA that is polymerase known as RDR6, which uses the cRNA as a substrate to produce multiple copies of dsRNA that is sharing the sequence of the original cRNA. That is also resulting the dsRNA is processed by this CL4 to produce more cRNA molecule, which can then spread further. And the short-range movement uses plasmodesmata, and the silencing signal correlate with plasmodesmata size, and the cRNA may move as single molecule or as dsRNA. So in this one, the cRNA move between plant cell via plasmodesmata, larger opening enhanced spread, and the exact cRNA form remain of a subject of the study. Evidence that the short-range spreading of RNA silencing uses a plasmodesmata comes from the observation that cells lacking functional plasmodesmata, such as God's share of the stomata, and it is not subject to silencing induced by neighboring cell. There is a correlation between plasmodesmata size and how far the silencing signal can spread, suggesting that it is easier for the cRNA to move through plasmodesmata with larger opening, and the exact physical form in which the RNA move between cells is still controversial. One possibility is the small RNAs hybridize together to form double-stranded small RNAs, which then move through the plasmodesmata. However, it is also possible that the individual cRNA molecules are the primary basis for the spread of the silencing. The last one, the cRNA enters phloem for the long-distance travel. It is systematic silencing resists viral spread and demonstrated via grafting equipment or experiment. So meaning to say, the systematic silencing enables cRNA to move through the vascular system, that is protecting distant plant region from viruses. The grafting experiment highlights its extensive protective reach. The effect of RNA can also spread throughout an entire plant, and a phenomenon known as systematic silencing. In this case, it is believed that cRNA molecules enter the phloem, possibly in combination with binding protein to keep them stable, and then move through the vascular system to distant region of the plant. Such systematic silencing can allow a plant to better resist viral attack, because once the portion of the plant is exposed to the virus, the silencing RNA can spread to the other region, and the systematic silencing can be demonstrated grafting experiment, in which a piece of 27 RNA response can induce silencing in the host plant after grafting being grafted onto it. And the systematic silencing is a striking example of the fact that cells are not isolated from one another as you, a plant undergoing, might think. Okay, so now let's proceed to the crispy or non-coding RNA in bacteria. The genomic region with viral DNA sequence separated by repeat and act as a genetic memory of past viral infection. CRISPR allows you to transcribe into a long RNA molecule, and ccRNA guide Cas9 to cleave a target DNA with the sequence homology. And the genome editing in eukaryotic cell, it enables also the mutation, insertion, and precise genetic modification. So we can say that within the genome of many bacteria, there are genomic loci containing a short DNA segment that are identical to sequences from the DNA of the bacteriophage viruses that is separated by a repeated DNA sequence. And these repeat loci are called the clustered regularly interspaced short palindromic repeat, or the CRISPR. The viral sequence are obtained from viruses that invaded the ancestor of the bacterial cell, and their presence in the bacterial genome continues memory of such prior attack. The CRISPR locus is transcribed as a long single-stranded RNA molecule, much like the pRNA prostitute, and then individual small RNA sequences called single guide RNA or CRISPR RNA are cut out and loaded into a protein called the Cas9. The Cas9 uses the bound sgRNA to locate the target and cleave any DNA with sequence homology to the guide RNA. Although this system evolved in bacteria to defend themselves against the viruses by cleaving the viral DNA genome, the CRISPR-Cas9 system has been engineered to work in eukaryotic cells by expressing Cas9 in a cell along with a suitable single guide sequence. It is also possible to create a double-stranded DNA break in the genome of the cell. When the cell tries to repair this break, it can lead to insertion of or deletion of DNA that is producing a mutant gene. And the CRISPR-Cas9 system does allow mutants to be made in any gene of the interest. Moreover, the repair process can lead to chromosome rearrangement or the insertion of the new DNA sequences that share homology with the region of the count, allowing the genome to be edited or modified at will, and the ability to create mutant and perform genome editing is virtually any organism using the CRISPR-Cas9 systematic has led to a revolution in genetic manipulation of cells. It is also particularly in organisms such as mammals and plants, where targeted genetic modification had previously been extremely difficult. Let us now proceed to another presentation, the other non-coding RNAs. So the transcriptome consists of all RNAs synthesized in a cell or organism. The mammalian transcriptome are vast, but their full purpose is unclear. Some consider it background noise or gene expression. So meaning to say, the DNA of a cell or organism constitutes its genome, and the protein it produces constitutes its proteome. The RNA that are synthesized by a cell or organism constitutes its transcriptome. Why is the transcription of the mammalian cell is so large? In other words, why does a cell transcribe all of these various types of DNA sequences? We do not know the answer to this basic question. According to one viewpoint, much of this pervasive transcription activity, it is simply the background noise that accompanies the complex process of a gene expression. Proponents of this viewpoint cite the result of the studies in mice, in which blocks of the genome that lack protein coding genes that have been deleted. It was found in these studies that healthy mice can develop even though they cannot synthesize the non-coding RNA that would normally have been produced from the deleted DNA sequences. And according to the contrasting viewpoint, much of the non-coding RNA being produced is involved in diverse regularity activities that have yet to be identified. And the supporters of this position cite results suggesting that this pervasive transcription is not random. Many of the non-coding transcripts display distinct and reproducible pattern of tissue that is specific distribution, and their origin can be traced to specific loci in the genome. Keep in mind that it has only been in the last decade or so that the existence of cRNAs or mRNAs and pRNAs has come to the light, and it is likely that other types of small cRNAs remain undetected. And not all non-coding transcripts are small, and a number of evolutionary are conserved long. So we have now reached the end of the presentation. Thank you for listening, and bye-bye.